Good afternoon one and all. So yes, in our previous session we have clearly dealt with two organelles which are included in endomembrane system, wherein the first organelle is what? Endoplasmic reticulum, both rough endoplasmic reticulum and smooth endoplasmic reticulum. Just a second. Yeah. So the first organelle is endoplasmic reticulum, both rough endoplasmic reticulum and smooth endoplasmic reticulum, where rough endoplasmic reticulum will be synthesizing out naive proteins which are inactive proteins or the proteins which are just synthesized, and smooth endoplasmic reticulum is involved in the synthesis of lipids. Okay, smooth endoplasmic reticulum involved in the synthesis of lipids. These naive proteins and lipids are accepted by the cis phase of Golgi bodies, which is cis phase or convex phase, cis or convex phase of Golgi, which accepts the naive proteins and lipids and proteins get packed inside the cisternae, which is lamellar structures. Okay, they are getting packed inside the cisternae and they are released out in the form of small secretory vesicles from the trans phase or concave phase of the Golgi. Concave, trans or mature phase of the Golgi. This trans phase or concave phase is towards the cytoplasm. Okay, this will be releasing out what? Secretory vesicles. These secretory vesicles are rich in hydrolytic enzymes. Hydrolytic enzymes like hydrolases, proteases, carbohydrases and lipases. Fine. Hydrolytic enzymes include hydrolases, proteases, carbohydrases and lipases. So these are being present in these secretory vesicles which have acidic pH. They have acidic pH. So these secretory vesicles are called as lysosomes. Fine. Secretory vesicles which are released out or synthesized from trans phase, concave phase of Golgi bodies. These are having what? Hydrolytic enzymes like hydrolases, proteases, carbohydrases and lipases. This has acidic pH. Okay, they are active in, these enzymes are active in acidic pH and these are lysosomes. Lysosomes are polymorphic in nature. Okay, they are polymorphic, means they change their shape, according to the molecule into which it is binding, or they change their shape according to the function. Those secretory vesicles which are just released out from the trans phase of Golgi, those which are released from the trans phase of Golgi, they are called as primary lysosomes. Okay, primary lysosomes, which are just released from trans phase of Golgi, which are rich in hydrolytic enzymes. These primary lysosomes, when they are attached with any digestive molecule or phagosome, this is said to be primary lysosome along with the digestive molecule or phagosome is said to be heterophagosome or secondary lysosome. Fine. And after digestion, post digestion, some undigested particles will be remained out. So those undigested particles are called as what? Residual bodies, which are being excreted out of the cell. And lastly, autophagosomes. Fourth type of lysosomes are autophagosomes. When secondary lysosome is adjoined with any dead cellular constituent or any dead cell organelle, for the lysing of that dead cell organelle or clearing of that dead cell organelle out of the cell, then it is called as autophagosome. Okay, secondary lysosome along with any dead cell organelle or cellular constituent. Fine. These are different types of lysosomes. So, with respect to lysosomes, what we have to remember, ma, lysosomes are nothing but the small secretory vesicles which are released from the trans phase of Golgi. They are rich in hydrolytic enzymes like hydrolases, proteases, carbohydrases and lipases, which are involved in the digestion of proteins, carbohydrates, lipids and fats. These hydrolytic enzymes will be active in acidic pH. Okay, and the lysosomes are polymorphic in nature. They will be changing their shape based on their function or based on the molecule to which it is adjoining. Primary lysosomes are the vesicles which are just released from the trans phase of the Golgi. When primary lysosomes are adjoined with digestive molecule or phagosome, these are said to be secondary lysosomes or heterophagosomes. And the undigested particles are residual bodies, autophagosomes, secondary lysosomes along with some dead cell organelles, that is autophagosome. Clear, right? Next. The last organelle in our endomembrane system is vacuole. Vacuole in a plant cell is said to be sap vacuole. Sap vacuole. Because this contains, sap vacuole contains all the water, sap and excretory products. Excretory products. Fine, they contain water, sap and excretory products. Vacuole occupies 90% of the cell space. Vacuole occupies 90% of the cell volume in a plant cell. And vacuole is surrounded by a single membrane. That single membrane is said to be tonoplast. Okay, single membrane, that is said to be tonoplast. Clear, right? So, it is called as sap vacuole because it has all these constituents. In vacuole, the pH is again acidic pH. It has acidic pH. The pH inside the vacuole is lower than that of the cytoplasm. So, those molecules which are digested by the lysosomes, be it undigested particles like residual bodies or the dead organelles which are digested by lysosomes, to deposit them into the sap vacuole, what transport is mediated? Important question. What transport? For example, this is the cytoplasm and these are the constituents which are formed after digestion inside the, after digestion by the lysosome. This is the vacuole. What transport is activated from the cytoplasm to the lysosome? As the pH from the cytoplasm to the vacuole. As the pH of the vacuole is acidic, the transport is definitely active transport. What mode of transport, ma? Active transport. Active transport because the molecules are being transported from low concentration to high concentration. Clear, right? From low concentration to high concentration. So, this is the activity of what? Sap vacuole inside the plant cell. This acts like complete deposit of the cell, where all the excretory materials which are present inside the plant cell are deposited into the vacuole. Clear, right? And next is different types of vacuoles we can see, like gas vacuole and food vacuole. Gas vacuole seen in the prokaryotic cells helps in buoyancy. Food vacuole observed in, okay, food vacuole observed in protistans like amoeba. Food vacuole in protistans like amoeba helps in engulfing food. Engulfing food. Fine. This is about gas vacuole and food vacuole. And vacuole which is present in euglena helps both in osmoregulation and excretion. Okay. This is about vacuole. As such, we have covered up total 4 organelles of endomembrane system. Let us once quickly revise the complete endomembrane system in one frame. As we have discussed, endomembrane system includes 4 organelles. First is what? Endoplasmic reticulum. Endoplasmic reticulum are nothing but, this is the nucleus and nuclear envelope. Endoplasmic reticulum is nothing but the reticular network of the reticular structures which are the extensions of nuclear envelope. These are the extensions of what? Nuclear envelope. Fine. So, above those organelles where you see, above those reticular structures where you can see ribosomes. In ribosomes also, larger subunit of the ribosome is binded to the endoplasmic reticulum by what? Ribophorin. Fine. Which subunit is binded? Larger subunit is binded, that is 60S subunit. Okay. And those which has ribosomes are rough endoplasmic reticulum. Those which does not have are smooth endoplasmic reticulum. Smooth endoplasmic reticulum will synthesize lipids and steroids if it is steroids like cholesterol, okay, if it is in the animal cell. Rough endoplasmic reticulum will synthesize proteins. So, these proteins and the lipids or steroids are accepted by which phase of the Golgi? They are accepted by trans phase of the Golgi, sorry, cis phase of the Golgi, which is convex. Okay, cis phase of Golgi which is convex. So, these are the lamellar structures which are cisternae interconnected in the animal cell, wherein cis phase of the Golgi is towards the endoplasmic reticulum and trans phase of the Golgi is towards the cytoplasm. Getting packed inside the lamellar structures, after getting packed, what happens? Synthesis of glycoproteins and glycolipids. Fine. And from trans phase of Golgi, small vesicular structures which are being released. Okay, these small vesicular structures are rich in hydrolytic enzymes. These are what? Lysosomes. Both the lysosomes, glycolipids, glycoproteins are being transported to the intracellular and extracellular targets. These lysosomes are deposited inside the vacuole, which is the sap vacuole in the plant cells. Okay, sap vacuole. Okay, this vacuole occupies 90% of plant cell volume. Fine. So, inside vacuole, all the materials which are being digested inside the cytoplasm are deposited in what way? Active transport. Remember, this is very important. They are deposited through active transport, that is by the utilization of energy from low concentration to high concentration. Fine, right? So, complete endomembrane system is clear to us. All the 4 organelles which we have learnt over here, be it endoplasmic reticulum, SCR and RER, Golgi bodies, lysosomes and vacuoles, all the 4 organelles are what? Single membrane bound organelles. They are placed under endomembrane system because their function is coordinated. Clear, right? Now we will look into ribosomes. These are non-membrane bound organelles which we will see in both prokaryotic cell and eukaryotic cell. So, these are the common non-membrane bound organelles which are observed in both the cells. Common non-membrane bound organelles both in prokaryotic cell and eukaryotic cell. In a prokaryotic cell, if you see, and in a eukaryotic cell, prokaryotic cell has 2 subunits, even eukaryotic cell also has 2 subunits of the ribosome. Larger subunit of prokaryotic ribosome is 50S, smaller subunit is 30S. Larger subunit of eukaryotic ribosome is 60S, smaller subunit is 40S. Okay. So, inside each ribosome it has what? RNA and proteins. Both the ribosomes, be it in prokaryotic cell or eukaryotic cell, ribosome is composed of ribonucleoproteins, ribonucleic acid will say, right, RNA, and this ribosome will have what? Proteins, amino acids and RNA. Okay. So, these are ribosomes are what? Ribonucleoproteins. They are ribonucleoproteins. If it is a prokaryotic cell, they have around 50 different types of proteins along with RNA. In a eukaryotic cell, they have 80 different types of proteins along with RNA. Clear, right? Any doubt anywhere, Vita? No. And next, so this is about ribosomes, and the ultimate function of ribosome is what? Protein synthesis. Protein synthesis is the ultimate function of ribosome. Now we have to discuss the location of the ribosomes. Okay, where are ribosomes present in a prokaryotic cell and in a eukaryotic cell? Ribosomes in a prokaryotic cell. If you consider this as a prokaryotic cell, fine, and this is the cell membrane. If we consider, attaching to the cell membrane, ribosomes are present, and ribosomes are present inside the cytoplasm. Those ribosomes which are present inside the cytoplasm, they are involved in protein synthesis. And those ribosomes which are attached to the cell membrane, those ribosomes are involved in the synthesis of proteins and translocation of proteins. Translocation of proteins. Fine, these are the 2 locations of ribosomes inside a prokaryotic cell. If it is a eukaryotic cell, where do you find the ribosomes? Ribosomes are found inside the cytoplasm. Ribosomes are found attaching to the endoplasmic reticulum. Okay, ribosomes are found over here, and ribosomes are observed inside the chloroplast stroma. Inside the chloroplast stroma, ribosomes are present. Okay, inside the chloroplast stroma, ribosomes are present, and ribosomes are also present in the matrix of mitochondria. Ribosomes are present in the matrix of mitochondria. Fine, right? So, here you have to be very clear that those ribosomes which are attached to the rough endoplasmic reticulum and those ribosomes which are present in the cytoplasm, they are 80S ribosomes. 80S ribosomes with 60S larger subunit and 40S smaller subunit. And those ribosomes which are present inside the chloroplast and inside the mitochondrial matrix, they are of 70S subunit, 70S with larger subunit how much? 50S and smaller subunit 30S. Just a second. Yeah, 70S ribosomes. Clear, right? Both the chloroplast and mitochondria, they are having 70S ribosomes, whereas the ribosomes which are present in eukaryotic cell cytoplasm and in the over the endoplasmic reticulum they have what? ATS type of ribosomes. So, and ribosomes which are associated, the larger subunit and smaller subunit are associated with each other, right? This depends on the magnesium ion concentration. Okay, the association of larger subunit over smaller subunit is depending on what? Magnesium ion concentration. More the magnesium ion concentration, more is the association. Less the magnesium ion concentration, less will be the association. So, this is mediated by what? Magnesium ion concentration. And then here monosome and polysome condition, both in eukaryotic cell and prokaryotic cell, you have to be clear with. Okay, monosome and polysome. Fine. Monosome is nothing but over the mRNA during the time of translation, over the mRNA, if only one ribosome is attached to it, okay, from 5 prime to 3 prime. In eukaryotes after 5 prime capain, what happens? Ribosome will get attached. So, mRNA with one ribosome is what? Monosomic condition. Okay, it gives only one protein expression, whereas polysome is nothing but single mRNA with many ribosomes attached over it, many ribosomes. So, post translation, after translation, this gives out polycistronic expression, that is multiple protein expression. This gives out polycistronic expression. Fine. So, eukaryotes monosome condition where single ribosome is adjoined with the mRNA. Prokaryotes polysomic condition wherein mRNA adjoined with many ribosomes and that gives out to be polycistronic condition. Monosome and polysome, we have to remember ma. Polysome condition is observed only in the prokaryotic cell, monosome in eukaryote, and polycistronic expression is observed only in the prokaryotic cell. And next non-membrane bound organelle that we are going to learn is centrioles, that we will exercise tomorrow. Centrioles and centrosome and cytoskeleton structures, microtubules, microfilaments and intermediary filaments, and we are also going to look into cilia and flagella structure, microtubular arrangement. Okay. So, that is all for today's session friends. Thank you.